Welcome back to Cancer as a Mitochondrial Metabolic Disease Basics. Fenbendazole is usually discussed in one of two ways. Some people call it a suppressed miracle cancer treatment, and others dismiss it as nothing more than a dog dewormer. But the interesting part is that fenbendazole is not alone. It belongs to a larger drug family called the benzimidazoles, and several members of this family have been studied for their ability to disrupt cancer cell structure, metabolism, and survival. So today we're going to separate hype from mechanism and ask a better question. What does fenbendazole actually target inside cancer cells, and how might those targets affect tumor metabolism? So without further ado, let's get into it. So we have looked at one member of this family already by doing a multiple part series on mebendazole. Today we're going to be looking at mebendazole's brother or cousin, fenbendazole, and see what kind of effects that it can have at the level of the cancer cell, and specifically the cancer cell's metabolism. So the first paper we're going to look at was published in October of 2024, and it was titled From Deworming to Cancer Therapy, Benzimidazoles in Hematologic Malignancies. And it says in the abstract, drug repurposing is a strategy to discover new therapeutic uses for existing drugs, which have well-established toxicity profiles and are often more affordable. This approach has gained significant attention in recent years due to the high cost and low success rates associated with traditional drug development. Drug repositioning offers a more time and cost-effective path for identifying new treatments. Several FDA-approved non-chemotherapy drugs have been investigated for their anti-cancer potential. Among these, antelmethic benzimidazoles such as albendazole, mebendazole, and fenbendazole have garnered interest due to their effects on microtubules and oncogenic signaling pathways. Blood cancers, which frequently develop resistance and have high mortality rates, present a critical need for effective therapies. This review highlights the recent advances in repurposing benzimidazoles for blood malignancies. These compounds induce cell cycle arrest, differentiation, tubulin depolymerization, loss of heterozygosity, proteasomal degradation, and inhibit oncogenic signaling to exert their anti-cancer effects. We also discuss current limitations and strategies to overcome them, emphasizing potential of combination benzimidazoles with standard therapies for improved treatment of hematological cancers. And in this publication, it had basically a graphic like this that had all of the family of these benzimidazoles. And as discussed in the very beginning of this video, we've already talked about mebendazole already, and we're going to be talking about fenbendazole today. And in coming videos, we'll be talking about albendazole, and I might also do a video on flubendazole as well. So this is just a look at all of the kind of therapeutic effects of these benzimidazoles within oncology, but also within other areas of medicine. And then it also looks at generally what they do. They're either going to decrease tumor growth, tubulin polymerization, oncogenic signaling, et cetera, and it's going to increase apoptosis, autophagy, cell death, differentiation, senescence. So let's look at specifically within this family of benzimidazoles at fenbendazole. So I'm going to start with this paper that was published in August of 2018, and it's titled Fenbendazole acts as a moderate microtubule destabilizing agent and causes cancer cell death by modulating multiple cellular pathways. And in the abstract, it says simultaneously, it caused mitochondrial translocation of PPD3 and effectively inhibited glucose uptake, expression of GLUT transporters, as well as hexokinase or HK2, a key glycolytic enzyme that most cancer cells thrive on. And I went ahead and pulled out of this paper the portions of the paper that were specifically talking about these metabolic pathways. And it says here, therefore, we examine whether fenbendazole, or FZ, impairs the enzymatic function of HK2, hexokinase, in cancer cells. An enzymatic assay for HK2 activity was performed after treating NSCLC, or non-small cell lung cancer cells, with fenbendazole for 24 hours. The results showed reduced HK2 activity in fenbendazole treated cells, suggesting that the inhibition of HK2 activity could be a causative factor for the reduced glucose uptake in fenbendazole treated cells, eventually leading to activation of apoptotic signals. It later says that succinate dehydrogenase, SDH, is a mitochondrial TCA, or tricarboxylic acid cycle enzyme, and is a known tumor suppressor gene. Succinate, a TCA cycle metabolite, is accumulated due to SDH downregulation. We'll talk about other reasons why succinate may be accumulating as well, and provides cancer cells with a growth advantage leading to tumor progression. Spectrophotometric assay, as well as histochemical staining, showed enhanced SDH activity in cells following fenbendazole treatment. So before I get into what these things are that is happening, as well as why they're important, I want to just look at what was found. Okay, so this was a table in this particular paper, and it gave certain results that I think are very interesting for us to look at and to study. So for example, in part B of this graphic, it's looking at glucose consumption compared to the control. And again, they're looking at two different types of non-small cell lung cancer models, this H460 and this A549. And you see here for the control that there is about 1 milligram per ml of glucose consumed in 24 hours by this cell culture. And when they added 1 micromole of fenbendazole, it showed a roughly 50% decrease in glucose consumption. At 5 micromole, we saw a roughly 75% decrease in glucose consumption. And at 10 micromoles, we saw a roughly 95% reduction in glucose consumption by these cancer cells. Now, then the next question was, how did this actually happen? And what percent change were there for other enzymes that were of interest for us? Specifically, hexokinase, HK, and we're going to look at this graphic here, this H, and then we're going to be looking at SDH activity in E here. And this table is built so it shows the 1, 5, and 10 micromoles if it's shown. So for this SDH activity, succinate dehydrogenase activity, we see that at control levels, compared to 1 micromole of fenbendazole, there's an increase of 80% of this enzyme. And at 5 micromoles, there was a 100% increase of this enzyme. Now, I'm going to explain why SDH and increasing its activity could be very beneficial to us. Stand by on that. But you're seeing that at 1 micromole, 80% increase, and at 5 micromoles, 100% increase. And then when we look at HK, hexokinase, this is an enzyme that is responsible for breaking down glucose in the glycolysis system. It's actually the rate-limiting enzyme for that pathway. We see that at 1 micromole, there's actually a slight increase of about 15 to 20%. That's actually not fantastic. But at 5 micromole, there's a decrease of 25% compared to baseline. And at 10 micromole, there is about a 50 to 55% decrease at the level of hexokinase activity when given varying concentrations of fenbendazole. What I will tell you is that it doesn't really line up too much when you see that at 1 micromole of fenbendazole, you're seeing a roughly 50% decrease in glucose consumption. And at 1 micromole here, there's actually a slight increase in hexokinase activity, which means there has to be something else going on. The authors didn't really explain what that could be, but that being said, it just doesn't line up. If you're decreasing glucose consumption by 50% at 1 micromole and you're slightly increasing hexokinase activity at 1 micromole, then there has to be some kind of other downstream effect or upstream effect that's happening that is leading to a consumption of 50% decrease, whether it's a downstream glucose enzyme or even a glucose transporter that is bringing glucose in at a lower rate. We'll look at that in a minute. But the succinate dehydrogenase is interesting because it's actually continually increasing it at increasing doses of fenbendazole. And there's actually a huge increase at just 1 micromole of fenbendazole. So let's quickly look at what the heck we're talking about. So succinate dehydrogenase is the enzyme that converts succinate to fumarate. So we're increasing the enzyme activity here. We're increasing the expression of this enzyme. There's more of it available to convert succinate to fumarate. So this is increased. That's why the arrow is going up. And then succinate, which is an oncometabolite, is going down. Now it's saying that the reason why succinate is built up in cancer cells is because there's a decrease in this enzyme. And this is a tumor suppressor gene, basically. What I would argue is that succinate is increased because succinic acid or succinate is the end product of glutamine fermentation. And we know that most, if not all, cancers are fermenting glutamine, aka they're converting glutamine into energy without oxygen or electron transport chain function into succinic acid. And that succinic acid is built up because there's heavy amounts of glutamine being fermented. That's why it's believed that succinate is built up. If there's a secondary reason that succinate dehydrogenase is downregulated in a lot of cancers, that even makes it worse. And why do you say that? Because it's not just an end product of this glutamine fermentation. It also makes up a significant portion of the extracellular acidification within cancer outside the cell. So succinic acid, among other things, are known as oncometabolites. They are metabolites that actually change the cell's behavior and gene expression. So we see here succinate leads to tumorigenesis here. It also leads to changes in gene expression. It also changes all kinds of other things like epigenetic regulation, redox balance, aberrant metabolism, genomic instability, post-translational modifications, pseudohypoxia, if you remember that term. Basically, the non-hypoxic activation or stabilization of hypoxia-inducible factors, a very key driver of the metabolic reprogramming that happens within cancer. It also leads to, here's succinate again, succinate leads to T cell exhaustion and leads to a more inhibitory kind of state that where the immune system is not effectively able to kill the cancer cells or attack the cancer cells. So succinate is what they call an oncometabolite. So what this paper is saying is that 1 micromole or 5 micromoles, they did in test 10 in that particular experiment, increase the activity of SDH by 80 to 100% at varying concentrations. So basically, as that enzyme increases, then the amount of succinate that's hanging around is being converted to fumarate more easily or more rapidly. So it's not hanging around having these oncogenic metabolic changes to the gene expression and to affect the extracellular acidification as well as the suppression of the immune system. Now, the paper also talked about how fenbendazole also was able to be combined with other metabolic inhibitors. So it says here, finally, we evaluated the effect of fenbendazole in combination with microtubule targeting drugs, Taxol, glycolytic inhibitor 2-deoxyglucose, and dichloroacetate, a pyruvate dehydrogenase kinase inhibitor, which acts by shifting the metabolism towards glucose oxidation over glycolysis. And what they did was they also looked at how the addition of fenbendazole, when added to a preclinical mouse model, how it affected tumor growth. So you see here that the control got some olive oil as a placebo, and then the treatment group got a dose of 1 milligram per mouse every second day of fenbendazole dissolved in olive oil. And they looked at the tumor mass after 12 days. And then they looked at the tumor weight compared to the control versus the fenbendazole group. And you see a stark difference in these non-small cell lung cancer cells. The next paper we're going to look at was published in September of 2024, and it was titled Oral Fenbendazole for Cancer Therapy in Humans and Animals. And it says here in the abstract, fenbendazole is a benzimidazole antiemetic agent commonly used to treat animal parasitic infections. In humans, other benzimidazoles, such as mebendazole and albendazole, are used as antiparasitic agents. Since fenbendazole is not currently approved by the FDA or EMA, its pharmacokinetics and safety in humans have yet to be well documented in medical literature. Despite this, insights can be drawn from existing in vitro and in vivo animal studies on its pharmacokinetics. Given the low cost of fenbendazole, its high safety profile, accessibility, and unique antiproliferative activity, fenbendazole would be the preferred benzimidazole compound to treat cancer. I'd say that's up in the air and debatable, but that's what these authors are saying. Is that actually fenbendazole would be the preferred one just because of all those reasons: safety, cost, accessibility, etc. This review focuses on the pharmacokinetics of orally administered fenbendazole and its promising anti-cancer biologic activities, such as inhibiting glycolysis, downregulating glucose uptake, inducing oxidative stress, and enhancing apoptosis in published experimental studies. So for this video, I'm going to specifically go over mechanism, and then in a subsequent video, I'm going to look at how fenbendazole tends to perform in either preclinical or clinical trials in humans. So it says here, fenbendazole is also known as this long chemical name, is currently used as an anti-parasitic therapeutic agent in dogs and other animals. In humans, other benzimidazole, such as mebendazole and albendazole, are used as anti-parasitic agents. Fenbendazole exerts its antiparasitic effects primarily in the anterior intestine by depolymerizing microtubules, inhibiting intestinal secretory vesicle transport. Fenbendazole binds to beta tubulin in parasites, causing microtubule destabilization and hindering tubulin polymerization. This destabilization disrupts cellular function, such as glucose uptake, thereby affecting the energy management of parasites. Due to its poor absorption by oral administration, fenbendazole is particularly effective for targeting intestinal parasites. In August of 2016, fenbendazole garnered global attention as a potential anti-cancer therapy following the complete recovery success story of Joe Tippins, who was diagnosed with small cell lung cancer. At the time, Tippins was undergoing a clinical trial for a novel anti-cancer drug. Meanwhile, under the guidance of a veterinarian, Tippins began self-administering 222 milligrams fenbendazole orally, along with vitamin E supplements, CBD oil, and bioavailable curcumin. After 3 months of self-administration, a PET scan revealed no detectable cancer cells in his body. Notably, Tippins was the only patient cured of cancer among 1,100 clinical trial participants. While the Joe Tippins case is compelling, it remains an anecdotal report. It underscores the need for rigorous clinical trials to validate the efficacy and safety of fenbendazole as an anti-cancer therapy. The anti-cancer activity of fenbendazole has been studied across many cell lines, demonstrating anti-tumor effects across multiple cancer types. Additionally, fenbendazole has shown efficacy against 5-FU, paclitaxel, and docetaxel resistant cancer cells. Compared to albendazole, fenbendazole was more effective against 5-FU resistant colorectal cancer cells, likely due to its intervention in glycolysis. Although fenbendazole exhibits promising anti-cancer effects, experimental studies indicated its poor water solubility has hindered its therapeutic performance. When administered orally, fenbendazole struggles to reach systemic circulation and, subsequently, the therapeutic levels necessary to impact tumors. Addressing pharmacokinetic limitations is crucial to repurposing fenbendazole as a cancer treatment. This review focuses on the pharmacokinetics of fenbendazole when administered orally and its promising anti-cancer biologic activities, such as inhibiting glycolysis, downregulating glucose uptake, inducing oxidative stress, and enhancing apoptosis. In addition, we evaluate the toxicity profile of fenbendazole and discuss possibilities for improving the bioavailability of the drug, enhancing its efficacy, and reducing potential toxicity. This comprehensive review aims to provide a detailed understanding of fenbendazole's potential as a repurposed anti-cancer agent and outline the necessary steps for its clinical application. And I pulled out basically the areas where fenbendazole seems to have activity. It specifically talks about, in this section of this publication, about how it affects GLUT transporters, specifically GLUT1, as well as hexokinase 2, which are two very important steps very high up in the glycolytic system. And so I pulled this kind of classic graphic that I've been using over and over again while we go through these videos, and I specifically pointed out with an arrow where fenbendazole is having an effect with GLUT transporters, the transporters that bring glucose from outside the cell into the cell, and through the hexokinase enzyme as well. That's where fenbendazole seems to have effects. My gut tells me there are several other effects, because when we looked again at this study where they were showing significant decreases in glucose consumption, and we saw big decreases also in hexokinase activity, but there's likely going to be a multimodal effect, because again, if you are getting a 50% decrease in glucose consumption and there's even a slight increase in hexokinase activity with the same micromolar quantities of fenbendazole, it makes me think there has to be other off-target effects that is just not being captured in these studies yet. And that's where more research probably does need to be done to know exactly where we're having these downstream effects at in the level of the metabolic pathways. But these are two critical steps, either glucose transporters or hexokinase 2, which is the rate limiting step within this glycolysis system. It later says that when administered orally at micromolar concentrations, fenbendazole induces cytotoxicity and effectively blocks cancer cell growth. Fenbendazole also causes oxidative stress and activates this MEK36P38 MAPK pathway, inhibiting cancer cell proliferation, enhancing apoptosis. So, everywhere I've ever seen MAPK, it ends up having an activating effect at upregulating glucose metabolism in multiple kind of graphics and publications. MAPK P38 is upregulating glucose transporters, hexokinase, and multiple of these downstream glucose related pathways. And it made me wonder, how is activating this pathway having an inhibitory effect on these cancer cells? And it made me kind of maybe better understand why at lower doses of fenbendazole, there was actually slightly increased activity in some of these glycolytic enzymes. And so the conclusion that maybe is possible is that it is a kind of a dose dependent response. So if you activate this pathway within a certain kind of dose and parameter, it may have activating effects on glucose transporters and other downstream metabolic pathways that are actually beneficial for cancer, potentially. This is a hypothesis. And at higher doses, higher concentrations, it may have an absolute opposite effect where it actually induces oxidative stress and has the opposite effect. And I think this is a to be determined kind of mechanism of how this actually works. But it does seem that even at 1, 5, and 10 micromoles, there are significant decreases in glucose consumption by the cancer cells. And also, there's significant increases in this SDH activity, which I do believe is a big deal. So if you like videos like this, please like, share, and subscribe. And until next time.